very, very pleased that you're all here and very pleased to welcome our guest, Mr. Jim Xu, who has a long career in the pharmaceutical industry as a statistician, and we thought it would be a great opportunity for you all, master's students in biostatistics and friends, to learn about opportunities for statisticians in the pharmaceutical industry. So thank you so much for coming and for making your slides available and everything else. Yeah.
Good afternoon, everyone. My name is Jim Xu. I'm a statistician for pharmaceutical industry for over 20 years, actually like 22 years. It's my pleasure to come here to give the presentation. So to help you to know what a statistician will do in pharmaceutical company, hopefully that will encourage you to decide to work for the pharmaceutical industry, to dedicate your knowledge and your effort to help people, to support the healthcare. So the title is a Statistician Life in the Pharmaceutical Industry. Table of contents, I try to cover different sections. The first one, I would like to give introduction to the pharmaceutical industry. And then I will give a brief introduction to the development cost and time for ethical drugs. We only talk about the ethical drugs, not the black market drugs, right? Only ethical drugs. And then I will give an overview of drug development process and also the important documents in the clinical trial. These trials will be the most important trials. If you become a statistician, you will dedicate a lot of your time to work on these documents. You will contribute to the document significantly. And then I will give some description about the statistician's role in drug development and the statistician's responsibility and skill set. Hopefully you can garner those skills during the school, and also try to practice. If you can have some internship, it can enhance your skill. And I will give a couple examples, so you can see some concrete examples to get a feel about what's going on in the pharmaceutical industry. So before I start, can I ask a question? What's your impression about the pharmaceutical company? Do you know anything about the pharmaceutical, or it's too far from you?
Lots of investment on R&D.
Yeah, that's right. Is yeah, go ahead.
It takes a long time to develop it.
Yeah, that's right. Or yeah, is the drug expensive or cheap, or some are cheap, right? Some are cheap, some are very expensive. We will talk about that later on. So pharmaceutical industry. The pharmaceutical industry is involved in the research, as one of the students said, non-process research, also the investment, and the development, manufacture, and marketing of products for the prevention, diagnosis, and the treatment of the disease. There are also other areas like for the cosmetic, like Botox, right? You can, I know that in Hollywood, the actors, actresses inject the Botox to keep young. But that's a consumer product. It's different from this one. So here we only, or say primarily talk about the prescription drugs and their over-the-counter medication. pharmaceutical companies aim to gather enough evidence to convince government agencies, doctors, and the patients that their drugs are safe, not harmful to the patient. Here, not harmful is a very conceptual language. Okay. No drug is completely safe because anything is toxic. If you drink water, you drink a 10 gallon of water in half day or in 1 hour, you got sick, right? Anything is toxic. It's a matter of money. So it's safe, not harmful to patient. It's not the exact language. You can say it's not really intolerable, meaning it doesn't cause a huge safety issue. Like for cancer patients, they're dying. Some drugs are saving their lives. There definitely are some toxicities, and toxicity could be very serious. Like for some drugs, it causes 50% of patients have neutropenia, and maybe 40% of other toxicity. Even like grade 3 is serious. But patients and their doctors have to balance the benefit and the risk ratio. So here, not harmful is not exactly correct or precise language, but that's a general is working. So we're trying to find the drug that is tolerable, also give patients a benefit. And effective here, effective. Improvement in patient's disease. Some may even cure the disease. Some of the disease I will talk later on in the later slide. They can cure the disease, even like a cancer patient. Ethical drugs, therapies, vaccines developed by pharmaceutical industry play critical roles in saving or prolonging patients' lives and curing disease, preventing virus like HPV here, human papillomavirus. That's a common virus. If healthy people like women can take that vaccine, they can prevent some sort of cancer. Also alleviating the disease symptoms to make your disease symptoms reduce. A simple example is just reduce your pain, like a pain medication. Right? After surgery, you have a lot of pain. If you are given some medications with opiate or other drugs, your symptoms can be significantly improved. Particularly critical for elderly people, because for elderly people like 80 years old, 70 years old, they have osteoporosis, hypertension, myocardial infarction, diabetes. They have bone weakness. If the fall, you can have a fracture. They have very bad consequences. But the drugs and the treatment may help them. This is one example. Probably some of you already read this news. The former U.S. President Jimmy Carter announced that his melanoma, one type of skin cancer, can spread to his liver from the skin. The cancer cell transport through the blood vessel blood to the organ, to his liver and the brain. Several years ago, the metastatic means already spread to other organs, like the Carter's would have been untreatable. So doctor gave him the radiation, and then consequently gave the Keytruda. This is a new drug, just approved a few years ago. And I gave you the very first 2 pages of the Keytruda. This is a drug product research. It's a very high-level description of what's going on here. And this is immunotherapy, the new type of drug. And this drug actually helped the president, former president, to completely get rid of cancer. So his tumor is gone. His tumor is gone. And there's no any sign of a tumor in his body. Even now he's still alive and no tumor. So this is really a very new breakthrough in the cancer research. Okay, last year the Nobel Prize has been awarded to two professors. One is Dr. Allison from Texas, University of Texas, MD Anderson Cancer Center, and another one is from University of Kyoto, Dr. Hanju. Yeah. They independently did research, and they found Dr. James Allison studied known protein, CTLA4, that has a function as a brake. So this CTLA4 can prevent the T cells. T cells are immune system, our own immune system. When there is a cancer cell or cancer cells, the T cell try to find them and then attack, destroy them. But this CTLA4 is trying to help the cancer cell hide, so the T cells cannot find it. But once you find the CTLA4 and try to inhibit, and the T cell will be empowered to find the tumor and then attack to get rid of the tumor. And that's the mechanism of one drug. And another one is from Dr. Hanju. His research is focused on the PD-1. PD-1 means programmed cell death. PD-1, programmed death. Because in the oncology, the theory is all the cells will die according to the program at the very beginning. And the birth is already programmed. Now, after the careful exploration, Dr. Hanju found the PD-1, and then PD-1 inhibitor was invented. So here, this is PD-1. PD-1, this is a T cell. T cell with PD-1, T cell cannot find the cancer cell. So cancers can grow bigger and bigger. But once you inhibit the PD-1, the T cell will be empowered to find those cancer cells and then destroy. It's similar here for CTA4. This is a T cell. It's trying to, make this blind. It cannot find the tumor here. But once this is inhibited, T cell will get a periscope or like a glasses like what I have here to see the tumor cell clearly and then can attack. So due to their breakthrough work, they were rewarded by the Nobel Prize. So who is Dr. Hanju here? Left one?
Right.
Oh, right one. You are right. Yes. Yeah, this is Dr. Hanju, this is Dr. Anderson. Yeah. So development cost and time for ethical drugs. I think one of the students told me, a lot of investment in the pharmaceutical industry. That's right. And if you read the news, every day you can see a lot of investment in the biotech companies. Yeah, millions, hundreds of millions of dollars, especially for those startups. Yes, the average cost for one drug to get approval is 2.6 billion. It's a lot of money And this is based on the series statistics from Tufts University. And the overall probability of clinical success, that means the likelihood that a drug entering clinical testing will eventually be approved is estimated to be less than 12%, or even lower. And for some drugs, it's much lower than 12%. 12% is high here. Some drugs extremely low, like Alzheimer and all. Most of the Alzheimer drugs fail. Even you find that it's so fascinating and your science background is so correct, but when you do the clinical trial, oh, it works so well in phase 1, phase 2. But when you go to phase 3, like thousands of patients, you fail. Then you find that it's almost same as taking placebo, the sugar pill. It's about the same. So you spend like billions of dollars and then you fail. So on average, it takes 10 years for a new drug to complete the journey from initial discovery to the marketplace. With the clinical trials on long, taking 6 to 7 years on average. So as a result, unfortunately, the drug is expensive. You also read a lot of news from media that people complain about the drug price, right? And for the Congress and the Senator representative, they're trying to cut the price. I think that's a good move. That's a good move. Yeah. But here you can see one example for Keytruda. Keytruda costs $150,000 per year, 1 year treatment for one subject, for one patient. So President Carter took this medication, then his insurance has to pay $150,000 a year. Since he has been taking this like a few years, the cost is pretty high. But if you can make a really blockbuster like Keytruda, you can also make a lot of money. Like Merck manufactured the Keytruda. It's anticipated they're going to make 9.3 billion annual sales this year. Yeah. And
So just a quick question, how long has this drug been always on the market for 5 years?
about 5 years. Yeah.
Are we expecting it to drop after 10 years being on the market?
Yeah, that's patent issue. Yeah, usually patent has 20 years to go from discovery to prove. Yeah, depends on how much time left. Yeah, they can have maybe another 10 years. And for the other drug, Humira, it's for the rheumatoid arthritis. I read the news. It's anticipated to make 20 billion this year. 20 billion for one drug sales. And I used to work on that, but early phase 1. That drug was developed by Knoll Pharma, Eisai Pharma. When I worked there, I worked on phase 1. I was a junior at that time. Yeah. And then Avalab purchased that drug. And actually, the sales price for the whole company was bought by Apple, including that product. Whole cost of purchasing the whole company was about 5 billion. And now they're making like 20 billion a year, or 1 year. Overview of a drug development process. After a drug is discovered by scientists, we will undergo the test in vitro. In vitro means in cubes, not in the living organ. In cube, culture dish, or whatever. And then it go up to vivo. Vivo is taking place in the living organ. And then will be in animal. It's called a preclinical phase, with the main goal of determining the safe dose for first in-man study in clinical phase 1 and assess product safety profile. On average, only 1 in every 5,000 compounds that enters the drug discovery to the stage of preclinical development becomes an approved drug. So 1 out of 5,000 compounds on average will be approved. And a lot of startup biotech companies, they just fail. Most of them fail. So this is like a flow chart. You start from here, stage 1 drug discovery. You have chemists working in the lab, and the scientists also have their rationale, they work together to identify the compound, and then go on to preclinical, so in animals. In animal data, they try to find the safe dose that can be extrapolated to human beings. Yeah. And FDA specifically issue guidance. It's called estimating the maximum safe starting dose in initial clinical trials for therapeutic in adult healthy volunteers. This gives the formula, like how to extrapolate animal data safety and safe dose to the human being. They say one of the most popular ways is to use conversion based on body surface area, and then convert by some numbers. If you are interested, I can leave this one to everyone. You can make a copy. Yeah, you are welcome to read, but don't spend much time on this one. Yeah, just for your curiosity.
And these are all online, right?
Yeah. I can also give the
the
link.
link would be.
Yeah, I can give the link.
Yeah.
So the chance usually takes a lot of time. You see here, 4.5 and 1 year and 6.5. Well, a lot of time, to get approval here. And the phase 4, stage 4 is a licensing approval. It takes 1.5 years. So when you become a statistician in a pharmaceutical company, you could be assigned to work in preclinical, meaning you will handle the animal data. You will not see human beings data. In human beings data is called a clinical phase. Clinical means only in the human being. If you work in the clinical phase, that's most of us statisticians do. Very likely you will be working in the clinical, not in the preclinical. Yeah, preclinical don't need that many statisticians. But for the clinical, it is not of a statistician. Yeah. And so we have a phase 1. It's a small study. Start with like 20 patients, could be like up to 100, usually not up to 100. Yeah. And the first study, you have to try to establish a safe dose. That's called a maximum tolerated dose. It's based on animals data. As I said, you use some conversion factor to convert the animal safe dose to a human being. But that's just a raw conversion. It's not guaranteed it's safe in human being. So you have to do other safety assessments to make sure you find the true maximum tolerable dose in human being. Yeah. And actually for oncology, you can simply use a 3+3 algorithm. It's pretty straightforward. 3+3. You first define what's the dose-limiting toxicity, like a grade 3 neutropenia, grade 3 hypertension. You define that. As long as patient hit one of the events, it's considered as toxicity. So you first enroll 3 subjects. If one of them has an event, end of the pre-specified event, you enroll another 3. If more than one, like 2 or 3 subjects have the event, then you say, no, stop here. You have to go to lower dose level because dose level cannot be the one you are taking. So you have to go to lower dose. Okay. So if only one has a toxicity, you enroll another 3. If among the 6 of them, only one, the original one, has a toxicity, you say this dose is safe because it's 1 out of 6. If it's a 2 out of 6, it's 1/3. 1/3 is like a 33% chance. It's kind of high, so you don't want that. You have to go down. So that's simple. For oncology, we also use the CRM, the continual reassessment method. It's sort of a simple application of Bayesian. Yeah, we use that also. So this is a very general description, but it gives an outline. So, and then once you find a good phase 1, the dose, you will do the PK. It's pharmacodynamics. You do the concentration. You get a sample, blood sample. You do the concentration. You try to see what's the time to the maximum concentration and what's the time to the steady state. You use UDA to describe the ADME. A means drug administration. D means distribution, and M means metabolism, and E means elimination. So you describe the process. So you will know how much of drug can be taken in your body. What's the bioavailability, or what's the concentration? You get a full description of your drug's behavior in the body. So about 70% of the drugs move to next phase. Phase 2. Phase 2 is usually for the efficacy, also the safety. You have a larger number of patients, so you can test your efficacy hypothesis, but not necessarily in very confirmatory way. It could be exploratory. As long as you see some singular, you say, okay, this is okay. I can pursue the phase 3. I have much bigger trial, and then I can try to show the superiority or non-inferiority, or whatever you define. Because your statistical power depends on the difference you try to detect, or hazard ratio. Right? And that if the hazard ratio is quite small, that requires not of number of events, also not of huge sample size. So that is usually not done in phase 2, but moved to phase 3. So phase 2 usually gives you some proof of concept. Also helps you to describe the dose response curve. You want to give different dose to see whether or not the effectiveness has reached the plateau. The dose response curve is like this one. It's non-decreasing, it's like an increasing. Somehow like logistic or Emacs model. You need to fully describe that because FDA wants that information. And then once you see the good signal of effectiveness of your drug, also the tolerable safety, you move to phase 3. The chance of going to phase 3 here, 33%. Do we have?
Yeah.
Do we have about?
Well, I don't know. I mean, how long do we have the room at home?
We do have.
And do people need to leave exactly at 3, or can you Day. So why don't you, we started late, why don't you, plan to take till, 3:15 or whatever,
Okay.
whatever you need.
And the ribbon's good until 4. So
Okay, yeah,
Okay.
yeah, so, I think, yeah.
So
Try
phase
to aim for 3:15.
Okay.
Yeah.
So phase 3 is large. You pivotal study. For normal submission, FDA requires 2 pivotal studies. They want you to show independent evidence because only one pivotal study even shows significant outcome. It might be due to some random effect, some other reasons. So FDA wants to know your result is really robust. A lot of times they ask you to do 2 phase 3 studies, both to show the significant outcome. But for oncology, usually they don't because it's hard to enroll that many patients. Also takes a long time to enroll. So they don't do that. Also for some large cardiovascular trials, they enroll like 10,000 subjects. You can't ask them to do 2 trials. It takes forever to enroll like 20, 30,000 patients. So 25 to 30% of trial move to next phase, and that's phase 4, meaning they get approval. This is a very general information from FDA's website. Try to cite some critical information, but this depends. For oncology, this might not be true. For some area like Alzheimer, it's much lower than this one. Maybe 10% or 5%. Yeah, really depends.
Right, I know like dermatology, I think it's much higher.
Yeah. Yeah. The criteria is not easier to meet, but for other disease, it's very difficult. For neuroscience, very difficult. Yeah. Phase 4 is called post-marketing. So usually company has a medical affair to handle this one. But statisticians still work on phase 4. This is mainly for the publication or for expanding your drug indication. For example, your drug has been approved for this type of disease. If you find it may work for another indication, you could do one. And if it really works, you could file for your new application. You can update this label. This label, you see here. Let me see. First one. Left-hand side, you see the indication. How many indications here? Melanoma, first one, right? And then they have others, non-small cell lung cancer, and then others. So the phase 4 may help to add more indications here. Okay, and then how can the drug get approved? First of all, before you start the clinical trial, even phase 1, before you start the first study in phase 1, you have to file. You have to file to the FDA and the investigation of new drug application. You have to give all the available information, like animal data and the in vivo data to FDA. And you have to provide a rationale and the protocol, clinical protocol. Let's say your phase 1 will start with study 101. So you have to provide your protocol, study protocol, writing back, scientific rationale, and safety, and everything you try to plan. You try to achieve the safety, everything you have to write into protocol and submit to FDA. FDA will say whether or not your drug should be investigated. If they find that this is really toxic, you can't apply in human being, then you cannot make a drug. You cannot make a drug and market in the marketplace. So they want all the information. And then FDA will review your drug. This can be put actually after other slides here. I will come back. Okay. New drug application. This is different from an IND. IND is an investigational drug application. A new drug application is usually after you finish the phase 3. You have the pivotal results. Phase 3, large pivotal trial, you already have positive results. And you feel confident, the safety is tolerable and the efficacy will benefit patients. Then you apply for this one. Yeah.
So if there's a disease that processes really fast for the patient, and will they still go through the phase 1 to phase 3?
I'm sorry.
I mean, if there's a disease that processes really fast, so the time to death is like really short. Will they still go through the phase 1 to phase 3?
Yeah. But we were talking about that. Some drugs can start the new drug application even in phase 2. If it's for the very serious disease, as you say. Yeah.
And also keep in mind there are different patients that are used. It's not the same patients that are used in phase 1, phase 2, phase 3.
Yeah, they're different.
And then in phase 3, it would take like years.
Well,
Yeah.
not necessarily. It could be short. If it's a terrible disease where the endpoint happens very quickly, it could be short. I mean, it may take a long time to accrue the number of people that you need, but the length of the trial might not. I mean, the length of time that any individual is followed might be short.
Yeah. Also, based on the situation in oncology, FDA would accept the surrogate endpoint. Like for the oncology, the gold standard endpoint, primary endpoint is overall survival, the OS. But overall survival takes a long time. You need to wait until a certain number of deaths has happened. It takes a long time. But you can use the objective response rate, like how many patients, what's the percentage of patients with a partial response and with a complete response. That's a surrogate endpoint. Yeah, FDA would take that as long as your drug is really critical for that patient population. So once you finish or almost finish your phase 3 or phase 2, as the students say, in very critical disease condition, then you can try to apply the new drug and submit a lot of information. I have been working on this several times. It's very, very intense. My kids know that no evening, no holiday, no weekend. Yeah. Work on. But this is the most important thing for the pharmaceutical company. If you miss this one, your drug will be rejected, then you don't make any profit. And all the investment will be in vain. So this is very critical, very critical. So you have to put your proposed labeling. This labeling, I only print the first page. Actually, this labeling has about 70 pages. 70 pages only give you first book to give you a sense of what will be covered here. Proposed labeling, safety, and drug abuse, patent, and any data from studies that may have been conducted outside the U.S. Institutional review board compliance information, descriptive direction for use. Here, yeah, as you asked, you see here, speeding the availability of drug that treat serious disease. Yeah, it's possible if the drug is really good, effective, for some sort of serious disease, you can apply the priority review, breakthrough therapy, and you don't have to go to phase 3. Phase 2, if you have a very good data, FDA will approve. This is not a typical way, but it happens. It happens. I think Keytruda itself went through this path.
There's also, maybe it was one of those, but there's like a conditional approval, right?
Yeah, exactly. First, like what I said for the surrogate endpoint, even you do the interim analysis, only when part of the trial is complete, let's say 70% of the events happen, you do interim analysis, you find the results pretty promising, you submit, and FDA could improve, but that's conditional. And even in the labeling itself, they may say this approval is based on the interim. It's conditional.
And so they can take it away if the results don't pan out again, and they've done that.
Yeah, exactly. Once you complete your phase 3 study, and you have to show your true primary endpoint. For oncology, it's overall survival. You have to show your overall survival is significant. If you don't show that, then they withdraw. Then this one, if this doesn't work, they cross out. You have to update this label. Then this indication will be removed. Okay. These document, these few documents are very important. You will work from you enter the pharmaceutical company until you exit the pharmaceutical company. So that's why I put them here. They are so critical. The clinical study protocol, it describes how a clinical trial will be conducted. It has objective design methodology, statistical consideration, and per FDA guidance, you must put this information here. So if you compare the study protocol from one company to another one, their sections and the titles almost exactly the same. They have to follow FDA guidance. So it will be like a page, background, objective, purpose, study design, and they have a selection and exclusion of subjects. What type of subject you want to enroll. You have inclusion criteria, exclusion criteria, the treatment of a subject, what medication you give the patient. Is that for oral or injection or whatever, or topical. And for statisticians here, you see assessment of efficacy and assessment of safety, discontinuation of a study statistic. You will work on this one for sure, for this one, also this one. You will also work on this assessment of safety because you have to provide all the analysis results. You will write the shelf, I will show you later, and give to programmers. You supervise the programmers. And the programmer will write their code, read the data, and produce a table you pre-specify. And you give to the clinical team, like physicians, and a safety officer, and even other team members. And statistical analysis plan. This is what you need to work on. And a higher level, probably you don't need to write these. You just supervise the more junior statistician to do. But in the beginning, you need to learn and practice to get a lot of experience. This is a very important document. FDA will question you if you make mistakes or something unclear. You have to write information here. This is one template because I cannot give you anything I have worked. It's confidential. So I give this from a public domain as an example. Yeah. You will cover these topics. You even specify what's the imputation method. If we have the missing data, if the data is missing, how you impute that one? How to calculate this variable? It's from randomization to the event date or from another date. You have to specify. Case report form. This is used as either paper or electronic questionnaire to ask the patient. This will be done by investigators. So they will record all the information we pre-specify. So they will collect a report and put into the database. And then as a statistician, you can analyze. And also table listing shells. This shells means you will, before programmers produce the output, you have to create a template to let them know what you want. And you also be able to specify the methodology. Some clarifications you be able to provide in the footnote, so programmers know what to do. Otherwise, sometimes it could be very, very confusing. You have to specify in the footnote. And the clinical study report, this is very important. For every single clinical study, once it's finished, you have to put all the results into here or table listing in the appendix. But the main body will be the description of the study outcome and the interpretation and your conclusion. And per ICH means International Conference of Harmonization, is E3. It must be in this format. Must be exactly in this format. So, what's a statistician's role in drug development? In 70s, 80s, statistician is more like a consultant, calculate sample size, and not major impact on the analysis and trial interpretation. But in 90s, statisticians start taking the role of reporting safety and ethics and becoming integrated into clinical development team. Since 2000, statistician become more strategic, moving from a statistician working in drug development to a drug developer with a statistical background, playing the important role, including but not limited to the following: design drug development plans, adopting and introducing innovative methods, and the protocol writing, statistical expertise to strategic and decision making, such as a go and no-go decision at the end of phase 2. Because at the end of phase 2, you have to decide whether or not your result is good enough to give you a good chance to succeed at the end of phase 3. So you have to decide whether or not you want to go to phase 3. Because phase 3 is very expensive. And statistical advice and support to clinical manufacturer process and operation. And instructing programmers to analysis production. Also, you need to validate the analysis results. Because programmers gave you results, some of them are wrong. I have to tell you this one. You can never trust everything programmer gave to you. You must validate. I have done many clinical trials, if not 100. I have not counted. If not 100, at least I can say many things. Every single study, you find the issue from the deliverable, from the programmer. So you need to be very careful about the quality. Once I found there's no any error. I was pretty surprised, and I found that simply use exactly same program to run the proven and to produce the table. Okay, they are correct, but they still made a mistake in the update, the cut update. So results still are somehow wrong. So interpretation of clinical study report writing, defending statistical method, and the results to the regulatory agency. Like FDA may question you, why you do this, how you interpret. Let me say, this is not valid, you have to defend. So you have to have a good knowledge of statistics and publication. Statistician's responsibility, you have to dedicate to the clinical trial design. Design is very important because clinicians, they have a general sense of disease, but they may not have the best sense of analytical aspect. In order to show the superiority or non-inferiority, you need a very careful calculation about the sample size, and you have to make sure your assumption for sample size are correct, and your best knowledge are good enough. And writing the statistical section of the study protocol, as I introduced in the protocol, is a very important one. You have to give details. And also reviewing other section of our protocol. Yeah. Working closely with the clinical team to design case report form. By the way, you need to develop a good interpersonal skill to become a good team player. That's very important. If you are not good team player, you will be kicked out, no matter how good your statistics is. Because physicians don't really understand statistics. If you cannot explain statistics in plain English, they get confused. They say, "Oh, you can't make us understand what you do." Then it's not good. And the statistical analysis plan templates, as what I say, supervisor, programmer, and working close with data management, interpreting study results, and defending the statistical methodologies, addressing questions from regulatory agencies, validating key study results. This is what I said. You have to validate, unless someone report to you will do that job. Otherwise, you cannot feel safe about the results. You have to validate. Otherwise, because when you send something to FDA statistician, they may validate. And if from the product they come back to you, and your company come back to you, you have to take the responsibility. Presenting study results to outside people working intensely on new drug application. Yeah, this is what I said. This is very intense. And these terminologies are probably too far from you, too conceptual. But when you work for the pharmaceutical company, you find them so important, so important. If you are able to work on the ISS, ISE, you are lucky, because this is almost last step for the drug to get approval, almost there. A lot of companies won't go that far. For most of drugs, they won't go to that stage. Only when you finish your pivotal study, have very good outcome, you do the integrated summary of the safety. Try to improve for standardization and this simulation, collaborating with outside companies, attending training workshop, and learn more new stuff, gaining necessary clinical knowledge, familiarize with regulatory guidance, providing expert input for publication, mentoring others. This is for more senior people. When you first join, you are the mentor. And the statistician's skill set, a successful statistician in the 21st century must have the following: the broad statistical technical skill, I learned from some of the students, also from Dr. Kapinski. Yeah, you are offered a broad range of statistical course. I think that's very good. Yeah, so you can try to choose as many as you can, but consider the study life balance. Yeah, but the more you pick, probably the more you will get a benefit when you work. The ability to learn appropriate clinical information from a specific therapeutic and disease area. So if you don't have a clinical knowledge, when you design the templates for analysis, you could be mostly wrong because you don't have relevant knowledge to analyze what should be done. So you need to have the clinical knowledge. Ability to review, interpret, synthesize, and communicate results from external and internal source. Ability to bring the lead expertise to the team while collaboration with internal and external experts. Ability to understand the problem and questions. Articulate the problem in clear, non-statistical terms. That's what I said. Yeah. So please try. As a student, when you discuss, try to use some plain English. Yeah, not so technical. When it needs to be technical, yeah, you have to use technical. But if you can use some plain English, that will help you to explain to clinical team. Excellent collaboration is very important. Professional conduct, yeah, is quite important.
In every job.
Yeah.
Whether you're in pharmaceutical companies or
Yeah.
anywhere, of course.
Yeah, exactly. In addition to get good statistical leader, the following also need mentoring, yeah, and be a good role model, yeah. And instilling a culture of continuous learning, making operation aspects of reporting a non-issue. Yeah, you can solve the problem, make that easy. And solid knowledge of following is required for most of the statistical tests in pharmaceutical companies. Like linear model, I forgot to put non-parametric. Non-parametric will be used, but not that much. And so you need to know Wilcoxon, right? Rank-sum, or even non-parametric and COVA, it could be used. But you don't need a very sophisticated non-parametric. Linear model, linear, nonlinear mixed model, generalized linear model, statistical inference categorical data, survival data, methods for missing data, and multiple imputation. This has been used many times in the pharmaceutical now. But I heard 20 years ago, it didn't take this one. But now they take. So excellent programming skill in science, very important. R is for simulation. And the software, SAS most important. And inquiry is and PASS and the Word, PowerPoint example. So here I give two examples. One is one pharmaceutical company planned a large trial, like 10,000 subjects for cardiovascular disease. It's a time to event, so it requires a lot of number of events. But they were not quite sure if the assumption is correct. So they did an interim analysis, and the interim analysis from the sample size was not enough. So they increased by 3,000, and then so they were able to establish a superiority. But the example shows without the interim analysis, they would most likely fail this one. Now, 3,000 is critical, very important. So statisticians played a very important role. The interim analysis, planning, and analysis for sample size reassessment. And another one is just some samples, how the results were looking like. This is actually from one of the table from the labeling here. Yeah, you can see what will be presented. Mostly this is for safety, the adverse reaction. This is actually just this is word class, and this is paper term, and it's summarized by percentage. All grades include grade 1 up to grade 5. And then they have grade 3 to 4. And then they have a lab, abnormality. They have all grades, grade 3, 4. And ethics, they have OS, overall survival, and how many person deaths, hazard ratio, and what's the p-value, and PFS, and so on. And best response. This is the overall response rate. This is the curve.
I think we should finish up, so if you could maybe just highlight the...
Yeah, we are done.
Okay.
We're done. Yeah, okay. So this is the last one. This is just a figure. Yeah, I think this is just some example. Yeah, I think I'm done with the presentation.
So anyone who needs to go should feel free to go, but anyone who has questions, we can stay around and you can ask questions about working in a pharmaceutical company or any more details about your role as an incoming master's level statistician or anything you want to know.
Thank you for your presentation. It's very informative and helpful because after graduate, I'm really interested to work in a pharmaceutical company. But my question is, when I apply for a job, the job description always says you need to have experience with the FDA regulation or the, I don't even know what's the CDISC thing. Or CDISC.
Say that's for the data. Yeah,
Yeah,
that's
so
for the data.
because we're students, we don't have such experience, or so I don't know how can I get that experience?
Yeah, I see. First of all, you won't have experience working with FDA as a student, right? For the CDISC, I can give you some information. Yeah, you can read, and I have some knowledge about it. Yeah, I can give you some help. But that's mostly for the programming, for programmers.
Yeah, because I searched the job title with the SAS programmer, they always says those requirements. So do you have any recommendations for like what kind of job title for entry level is more suitable for a new grad?
Yeah, they have statistical programmer. Yeah, you usually have that.
Is that different from statistician?
It's different. It's usually statistics and programmers are in one department, same department.
Okay.
And statisticians usually they have some type of role of supervising the programmers to provide the request and the specification of a deliverable to programmers.
So what would someone who's just graduated with a master's in biostatistics, what would they apply for? What position?
I mean, you can apply programmer. You can also apply the statistician. Yeah, you can apply both. Yeah. But depends on your intention for your career growth. Programmers support the statistician by working on the tables, listings, and the figures, and provide to statisticians. And the statisticians share this information with the clinical team.
But somebody straight out of a master's degree could be could apply for an entry-level statistician position.
Yeah. Also in marketing, like New York City has a lot of marketing companies, like American Express and others. Yeah, they hire junior statisticians. if they have positions.
So how proficient do students need to be in SAS? So let me just give you the little background. So here, many of the courses are taught in Stata, using Stata. Some are taught with SAS. Sometimes students have a choice of using SAS or Stata or R. But how important is it that students be very proficient in SAS?
For pharmaceutical, I think SAS skill is very critical.
Okay.
Even you are not a programmer. If you are a programmer, you must be very good at SAS. You don't need to know anything about Stata or even R. But you have to be excellent in SAS programming.
So are there online courses? Or?
There are. I think there are some trainings. In SAS. Yeah, there are some.
So maybe could you send? I'm going to get a lot of information from you, but
sure.
Okay, I will follow up with that. Okay.
Yeah.
Okay. Other questions?
I know a lot of job postings, they were just turning off, like saying that if you're sponsored under OPT, they won't get you.
Oh, you mean the sponsorship?
Right, like a lot of them, like they would not sponsor like entry level positions. Rather, like if you are hired out in more like executive positions, they will more likely to sponsor.
Yeah, that happens now because immigration has more strict policy. It's getting harder. But you can always search. There are some companies that would like to go sponsor.
Some pharmaceutical companies do.
Yeah.
We'll do that.
Yeah. So when it comes to the design phase, I guess these statisticians have the power or the ability to decide what statistical methods will be used to analyze a trial.
Yeah. But there are also some standard ways to handle the common endpoint variables. So let's say for the survival analysis in oncology, if you want to analyze time to event, you always use a log-rank to compare the two curves, and then you use Cox model to either stratify or non-stratify to calculate hazard ratio. It's almost commonly used.
Although I thought with immunotherapy, companies even are moving away from log rank, that it might be more powerful to use some other test, or some test that weights differently more toward the end of the right tail of the curves.
Yeah, there are, but at this time they have not really adopted that yet.
Okay.
They are probably
Thinking about it.
Yeah, thinking.
So studies do biostatistician has the ability to actually get involved into like a laboratory side of the research?
No. That's for scientists.
Right.
Scientists are chemists. Yeah.
But don't they have, some statisticians work in preclinical. Don't
Yeah,
they?
but it's a small percentage.
Yeah.
They don't need that data. But when you go on to clinical, so many clinical trials. But for preclinical, it's so narrow.
Right.
Yeah.
Right, and they probably use pretty much standard designs.
Very standard.
So maybe they have
very standard.
less
Yeah.
need.
Right.
But for the clinical trial, even data itself takes statistician so much time. And when I work on some paper study, like oncology study, basically, I had to validate some important points. Right? I wrote my whole program, could be like 20 pages of SAS code. I started from scratch, from very raw data, merge and very complicated. And I try to match what the programmers did. And sometimes I found that there are errors. That's why I say that you can always, not always trust what programmer delivered to you. always something you cannot find without writing book. You can never find the problem without writing book.
Yeah, could you give us some recommendations on, like, for example, how to explain our statistical results on a plain language? Since, for example, in academia, like, for example, our professor and people around us are all like know something about statistics, but for example, there's no way they could improve that kind of skill, since like everybody already know it, and also the academia are using the technical languages. So how to practice on that?
Okay, let's see. Let's consider example. One simple example. Let's say the drug for the weight drug, right? Let's say if the average drug after taking the drug for half a year is like 5 pounds or 5 kilograms, or 10 kilograms. Let's say 10 kilograms. And the physician would tell you, probably needs 20 subjects per arm to compare them, because this drug 10, another one placebo only 1. The difference of 9 is so huge. Why do we need like 100 patients? Yeah, you have to explain to them. You can tell them that the power is determined not only by the delta, the difference, but also by the standard error. Right? You need to explain to them. So it's 2 aspects. Actually, it's not simply the difference. You have to bring the other factor, like a standard error, to the team. But they don't know what's the standard error. You have to educate them somehow.
Sometimes showing pictures can help, showing what raw data look like.
Yeah, if you plot the raw data, you will say, oh, it's really massive, you cannot tell much difference. The mean difference is 9, but when you look at the scatter plot, oh, not much difference. Yeah.
So how long did it take you from like an entry-level statistician to actually be more proficient on all kinds of statistical skills? Because I know, like, even though we learn it in school, you might not be necessarily proficient. Let's say if you see a bunch of data, you may not be like able to know what kind of analysis we will do, and then like, the procedure, like the flow of analysis.
Yeah, it depends on your chance or how your manager will assign you to what type of work. If your manager always assign you to the phase 1 and not assign you to work on phase 3, or you never gain that important experience for the larger trial, for not more complicated situation. So you need to first demonstrate your potential by working hard and professionally to show your manager, you are good and you have the potential to do more complicated work. Yeah, then you could be assigned to higher level of work. Yeah, and graduate to even higher.
And also, how often do you guys use parallel linear regression? So which is like multi-level linear model, multi-level model for analysis.
Multi-level or next for the like Bayesian, right?
Right.
Yeah, actually, so far for clinical trials we never use. But for myself, when I do research, I may use that.
Well, if you mean, it depends what you mean. Like if it's like a mixed effects model, like a longitudinal model. So those would be used like in like Alzheimer's disease, when there are measurements at every 3 months.
Yeah.
That could be the primary endpoint. That could be the basis of the primary endpoint, like that particular type of mixed model.
Now FDA is not that open to very innovative method like multi-level analysis and Bayesian. But they're open to the mixed model. They even issue some guidance on using the Bayesian analysis and even Bayesian adaptive design. But they have not really adopted the multi-level.
Because I think like maybe different countries, because people have different lifestyle, different backgrounds, maybe the drug effect might be different among different countries.
So usually that's like a stratification factor.
Yeah.
So they do account for that for sure.
Right.
Yeah, that's right. You can, if you really predict there is like a region effect, and that effect is pretty big, then you can do the dynamic randomization, stratify by region and by other factors. So there could be some balance within that region.
Another question. I heard many senior SAS programmers can work from home or work remotely. Is that correct?
Yeah, that's right. Some programmers, yeah, could work from home, but I heard that some companies in California they hire some consultant from here. Then you can work from home here. You just call them, or they give you, computer. You can connect to their network.
But it has to be a senior title.
Yeah, because they can efficiently communicate with you through the phone. Yeah, if you are a junior, you need training.
Anything else?
Yeah, for example, for one drug, for example, for any drug, if they want to sell it in the U.S. or like even in some other countries,
Like
do they
China.
in China, for example, do they have to do phase 1 to 3 in both countries? Because I heard China has more.
No, they do phase 1 to 3 in U.S., but they included the patients
Okay.
from U.S. and from Europe, and then some from China, some from Japan, for example.
Okay.
So if you plan to get a drug approval through the Chinese FDA, it's called a CFDA, then you should include in those certain number of Chinese patients. If it's too small, if the data is not good, you may not get approval. So at the planning time, you have to specify about how many Chinese patients you want to enroll.
So the experiment gonna be among other countries, both countries, not individually.
It depends on availability of patient population. Usually they have in their mind, they have some proportion, like how many persons maybe from US, how many patients from EU, how many persons are patients from Japan or from China.
Okay.
So when you guys hand in the statistical reports to FDA, will you also give them the data so they
Yeah.
are going to replicate your
Exactly, yeah, you submit all the data.
Right.
All the data you have.
Right.
The animal data, the in vitro data, and in people, all the data.
So they're also on their side, they're trying to replicate, see their results is matching with what you
Exactly, yeah.
You give them their code also, right?
Yeah, you also give your code, all the codes to them. Yeah, so if you are a programmer, when you write a code, you have to document your program. You have to say what's the purpose of this paragraph or code. Otherwise, no one can understand.
Yeah, I'm curious about the responsibility of, as a statistician or biostatistician. For example, you do both design and also analytic part of
Yeah.
or some people do one part, some people do other part.
Usually, if you depends on you can do all the hands-on work. Yeah, most of studies do that.
Okay.
Great. All right. Well, thank you so much. This is very informative.